What we're going to focus on next is another process that can happen to DNA in the nucleus of a cell. We have learned before that DNA holds the codes to build all of your proteins. So we're going to take a look at how we can read the DNA and get the instructions for to build a protein and how that protein is going to be assembled in the cell. Whenever we're done today, you should be able to describe all of these learning targets that are in this third box over protein synthesis. Now, unfortunately, when I teach you about protein synthesis and what's going to happen to DNA, it looks really similar to what happens to DNA whenever it is replicated. And then students have a tendency to get these ideas of replication all confused with these ideas related to DNA and protein synthesis. So it is critical that when you go to study, that you pick up differences between DNA replication and what's going to happen to the DNA during protein synthesis. Again, these are just two processes that involve DNA, but replication has nothing to do with protein synthesis, and protein synthesis has nothing to do with replication. So let's start mentioning replication, and just highlighting this main idea is that the only reason why a cell needs to replicate DNA is because it got a signal to divide by either mitosis or meiosis. So when it gets a signal to divide, then the cell then goes from G1 into S phase, where it's going to copy its DNA. So again, replication only happens during S phase of the cell cycle. Protein synthesis is a process that relates to DNA, and it happens 24/7. So I want to highlight that all throughout a cell's life, and you can even bring it over here through mitosis. All throughout a cell's life, protein synthesis is occurring. Proteins do the different jobs for the cell, so we are making proteins every minute, hundreds of thousands of them, to do specific jobs for the cell, and it happens all throughout the cell's life. Replication only happens during S phase, and it's because a cell needs to divide. So protein synthesis is just a fancy way of describing the making of proteins according to those codes in the DNA. As I mentioned before, the cell is making protein all the time in all the phases of the cell cycle. We can call proteins different words. We can call this a protein, which you see here, or it also can be called a polypeptide. So this is also a polypeptide. So you can kind of interchange protein and polypeptide. And remember, a polypeptide, poly means many. We have many monomers hooked together to make this protein or this polypeptide. And the monomers that we have to link together, mono means one. So one of those circles right there is a monomer, is an amino acid. So let's indicate that the monomers of proteins are amino acids. So we need to link together hundreds, if not thousands, of amino acids if we want to make a single protein. There's two steps in protein synthesis, and you need to know these words: transcription and translation. They sound similar, and students will often mistake which one happens first, which one happens second. So transcription happens first, translation happens second. If you forget and you're trying to figure out during your quiz which one comes first, then I'm going to put them in alphabetical order. If you can alphabetize, you're going to see that transcription comes before translation. This diagram is just attempting to show you kind of the two big processes and where they're going to take place, and in general what's going to happen. And then we're going to dive in and look at the specifics of transcription and the specifics of translation. If I ask you to describe what's going on during transcription, then this is what you need to tell me. During transcription, we're going to copy the gene of a DNA molecule. That copy that we're going to make has a name. It's called messenger RNA, or mRNA. When we get to translation, this is what's going to go down. A ribosome is going to read that copy. It's going to read the mRNA. Those are the directions, or the instructions. And then it's going to build a protein according to the directions, the instructions. So again, transcription, as you see here, we have DNA and we're just going to copy the gene. So I want to highlight this section of DNA that codes for a protein is called a gene. That should not be new information to you. Sections of DNA that code for a protein is called a gene. We're going to copy this gene, and that copying is called transcription, and we're going to get a new molecule, the copy. So let's write, this is the copy of the gene. This copy is called mRNA. So transcription occurs in the nucleus. If we're going to copy DNA, we have to copy DNA where it's located in the nucleus. The second step, so again, transcription was the first step. Translation is the second step. Translation is going to happen out here at an organelle that we've already talked about, the ribosome. So we see the mRNA is going to leave the nucleus, because here is the mRNA now. It traveled out of the nucleus. It's in the cytoplasm. And this organelle has clamped down on it. It's shown much bigger than what it really is. Remember, ribosomes look like small little dots scattered throughout the cytoplasm, and there's millions of them because we have to make a lot of proteins to do the different jobs of the cell. So we say that translation occurs in the cytoplasm. You can also say translation occurs at a ribosome. You can mention both of them. But you need to remember transcription happens in the nucleus and translation needs to happen at a ribosome. Now I want to emphasize why we go through all this trouble making a copy. So imagine if you build a new house and you decide to design your own. So an architect is going to design your house for you. Those blueprints are going to cost you about $7,000, depending on your home. So let's say you now have a copy of your blueprints. You do not just take your blueprints to the construction site and leave them there, because someone can steal them, they can get damaged, they can get ripped, blown away, and so on. But what you do is you keep your original blueprints, and then you just make several copies of them, and you send the copy to the construction site. That's what I want you to think of when you think of transcription and translation. This is my blueprint of my house. I'm not going to jeopardize it. I'm going to keep it in the nucleus forever, but I'm going to make a copy of it, and I'm going to send the copy out here to the construction site where we're actually going to build a protein. So if you haven't figured it out, during translation, this is the protein that is being built, and each little circle is an amino acid, is a monomer. Now we're going to get into the specifics of transcription. So again, that first step of protein synthesis is transcription. Here's a good diagram of transcription. We are just going to copy, not the whole DNA ladder. That happens in replication. We're just going to copy the gene on the DNA molecule. One gene for a protein that the cell needs to make. And again, that copy of the gene is called mRNA. And you can see mRNA right here. Now, notice, we're just building off of one side of the DNA after it's been split. In replication, we unzip DNA and we copy both sides. But in transcription, we unzip the gene and only the gene, and we're going to build mRNA just off of one side of the DNA. And we'll always tell you which side of the DNA to transcribe. But we're not going to transcribe both sides. It doesn't happen. To summarize and go step by step, and make sure that you can then do transcription. Just remember, DNA is unzipped at the genes, or at the gene location. To unzip it, we have to break those H bonds. There is no helicase. So let's indicate that. This enzyme that is making the mRNA, you can actually see it right there. It is unzipping, it is adding some RNA nucleotides to that side of the DNA, and it's actually rezipping back up the DNA. So there's no helicase involved in transcription or protein synthesis. Now, this enzyme that is copying one side of the DNA, that's making this mRNA molecule, it is building the mRNA not out of DNA nucleotides, but out of RNA nucleotides. And you're going to see what I mean by RNA nucleotides, because you're going to have a pile of those in addition to DNA nucleotides at your lab tables. You're going to see that whenever you're matching up RNA nucleotides, there are no nucleotides that contain thymine. Instead, you'll see a new base that we haven't talked about yet, which is called uracil. You only find uracil on RNA nucleotides. Some of you maybe noticed that up here, when we were building the mRNA, there's no T's. Instead, there's U's. So anytime your pencil wants to write a T, if you're making RNA, you can't place a T in there. You have to put a U. So let's kind of go through what this is going to look like. Here's DNA, and it's going to unzip. And now we have it separated, like you see right here. We're going to make mRNA by copying just one side of the DNA. Let's go ahead and copy this side over here. And again, you can write A's and C's and G's. When you want to write a T, you can't. You have to put a U. So T goes with A, because I can write A's. But A goes with, you want to write a T, but we're making RNA. There's no thymine, so you have to put a U. C goes with G, A goes with U, A goes with U, G goes with C, and C goes with G. This is the mRNA molecule. After the mRNA has been built or transcribed, it leaves the DNA, and the DNA, as you can see right here, just zips right back up. So now that we've kind of gone through that process, again, it's just a visualization, another diagram to show you that this is transcription. So if you see something like this and you see something being made off of one side in the middle of DNA, you notice that there's U's, then you would have to be able to tell me this is not replication, this is transcription. The cell is trying to make a protein eventually. After the mRNA is made, you can see it leaves, and it's going to make its way out to the cytoplasm to a ribosome. And that's where the next step is going to take place. So before we go into the next page of our notes, you guys are going to pause, and you're going to go to the lab tables. You're going to build a molecule of DNA, a long molecule of DNA. You're going to section off an area, that's the gene, and then I want you to show me transcription. Now remember, you have two piles of nucleotides, and you'll be able to tell the difference between the two. One pile is the DNA nucleotides, and the sugar is deoxyribose. The other pile is going to be RNA nucleotides, and you can tell they're RNA nucleotides because the sugar is ribose, not deoxyribose. Make sure you keep your two piles separate, because if you mix them up, you're going to have to separate each and every nucleotide back into two different piles.